The Spin-off Podcast Network.
K-pop to me means more than just listening to music. It's learning to be myself.
The Spin-off's new documentary, K-Pollies, follows 3 Pacific youth obsessed with K-pop. In a one-off documentary, see what they've found in Korean pop culture and how they bridge it with their own.
When you start dressing, looking different, everyone side-eyes you. But in K-pop, they're just like, no, like celebrate yourself.
Watch K-Pollies today at thespinoff.co.nz/videos. Made with the support of New Zealand On Air. You're listening to Business is Boring, a podcast that reckons it's anything but. Business is Boring is brought to you by Spark Lab, offering inspiration and practical advice to help businesses find their edge. To hear more about Spark Lab, including details about the latest events, workshops, and business tools, visit sparklab.co.nz. And now, here's your host, Simon Pound.
You're listening to Business is Boring, a podcast that reckons it's anything but. Business is Boring is made by The Spin-off with help from Callaghan Innovation. Here's your host, Simon Pound.
With the onset of Covid-19 came a huge global effort to create vaccines. An amazingly successful one, with new breakthroughs in technology and approaches that we are still at the beginning of understanding just how they might change health and medicine. Vaccines went into development all over the world, including in New Zealand, where a local company is working to develop a new approach that may use a technology developed by a Palmerston North-based company called Polybatics. The company, CVC, brings together some of the top figures in local pharmaceutical, deep tech and life science commercial enterprise. The chair of CVC is the managing director of Douglas Pharmaceuticals, and the CEO is Dr Robert Feldman, who's had a career working at Harvard Medical School, Imperial College London, founding the UK vaccine company Microscience, among other enterprises, before moving to New Zealand as executive director of Pacific Channel, and after that, his own life science consulting company. To talk the science, the journey, and vaccine research, Dr. Robert Feldman joins us now. Tēnā koe, thank you for being here.
Good to be here.
Hey, so first up, tell us about your background working overseas in commercialising life science research. How did you get into that field and, be in a position to be starting vaccine creation companies?
Yeah, I start life in London, born and bred in London. I first trained as a doctor. I became a consultant and professor in infection, and particularly infections in newborn babies. That was at Hammersmith Hospital and Imperial College in London. And so that's where I first got excited and quite driven by the whole field of infection. I then, overlapping with that, I developed a academic career. And that took me to places like Harvard Medical School and University of Utrecht, in the Netherlands, where I did my PhD. And that was in infection and immunology as well. And I was particularly interested in an infection, a bacterial infection that kills newborn babies. And I was working back at Imperial College and was working on a vaccine that might help protect these infections. And so that's when I first got interested in vaccines. And actually then I kind of flipped and turned my academic research into commercial research. And this is where I formed the company Microscience, which was a vaccine company. It worked on a whole set of vaccines. The one I was talking about for this infection called Group B strep that kills newborn babies, but also meningitis and typhoid and hepatitis. It had quite a broad portfolio of different vaccines to try and help people. That was my first company. So that's how I got into vaccines.
What was it that attracted you to that field? Like, As an outsider, vaccines seem like this absolutely miraculous thing, being able to, help to beat and remove diseases and things that have, plagued humans. They just seem remarkable.
Yeah, well, that question's beyond me. What makes us tick? Why do I get interested in vaccines? Why does someone else get interested in drawing wonderful drawings? Why does somebody love doing sports? I don't know. What how different things drive us, but certainly my gig is vaccines. I love vaccines.
And tell us about how you came to be in New Zealand. What led you here?
Well, I could just say a plane got me here, but which it did. But this was during the global financial crisis, and after the Microscience, the vaccine company, I also started and run a number of other different biotechnology companies, all in the infection field. And then the global financial crisis came along, and the UK was really tough in the field of biotech, particularly tough. The venture capital industry was in a bad shape before the global financial crisis in the UK. And so when the GFC hit, it was terrible. And VC money, the venture capital money, is the engine that drives the biotech industry, and there was no money. And at the same time, big pharmaceutical companies in the UK were disproportionately laying off people there. And so there were a lot of very skilled people chasing no money. And somebody offered me a job here in New Zealand. So I did quite a lot of homework and said, should I come, shouldn't I come? And I came. And that was nearly 11 years ago. So that's how I arrived.
And that job at Pacific Channel, which is was a great outfit that's doing a bunch of work in commercialising deep tech. Tell me about the work that you did there and what it was like to be kind of on the other side of the company and helping to move companies along rather than start them yourself.
Well, I'd done it before because I'd also worked for 4 years in venture capital. I set up and run a venture firm in the UK, which was a subsidiary of a US firm. So I'd been on that side anyway. So that wasn't new to me. But it was great working for Pacific Channel. There was rather less human biotech than I had hoped there would be, or maybe even was led to believe. But so my skills maybe weren't used optimally because in fact the greatest success I had was creating and causing to exist a company called Engender, which is an ag tech company. It wasn't, and I was kind of winging it. I know how to set up companies and I set up the structure, but I found the technology, but it's not really my true skill. So it was a good time at Pacific Channel. It got me over here. It got me the networks, and after leaving Pacific Channel, I could concentrate on what I think I do best, and that is human life sciences.
Yeah, and then being here and then seeing, COVID-19 hit the world and the vaccine efforts start. Tell me about pulling together kind of the team that started CVC. And were you looking to kind of get back into vaccines, or was it just that there was this enormous global challenge to help try and counter?
It was a year ago now, wasn't it? And I think we're already forgetting what it was like to live through that. It's already beginning to be a bit distant memory, but we were all ignorant. We were all deeply ignorant. We didn't know what was going to happen. We didn't know how worried we should be, but we were all pretty worried. We didn't know just how, the effects on the globe. There were just so many questions at that point. And Covid-19 Vaccine Corporation came out of a conversation I was having with a colleague of mine whose name is Andy Herbert. And I think I'm right in saying he's probably the only other person other than myself in this country that has had commercial experience in the development of infectious disease vaccines. Maybe I've just left someone out, but there's certainly not many of us. And we knew each other, and we had a chat, and we asked each other, is there anything that we can do that materially adds to the huge, well-funded effort that was already going on around the world? And we thought, well, yes, because there were so many unknowns. We just didn't know how easy it would be to make a vaccine, and we didn't know what kind of delivery mechanism would be good for a vaccine. And we identified this New Zealand technology that was developed at Polybatics in Palmerston North that actually lent itself quite well for a vaccine delivery mechanism that was different from what everybody else was doing. So we said, well, here is a technology that nobody else is using. Maybe it's good, maybe it's not good. But let's try it to see if it's going to be able to produce, good immune responses to COVID-19. And at least it's something different from what everybody else is doing. There's no point in repeating what somebody else is doing because whatever we do here is not going to be as well funded as some of the other projects around the world.
And your point there that, we didn't know how scared we should be. Yeah, I really agree with that idea that, we are forgetting just how ignorant we were. And, now it's just, of course, there's a vaccine and people are being, skeptical and idiots about it. But at the time, I remember doing some reading around it. And, there was a very real chance that it wouldn't be possible to get a vaccine, right? Is that right? Like, just help us contextualize, like, just how much of a breakthrough some of these vaccines are.
I thought it was pretty likely we'd get a vaccine. What was unclear in my mind was how effective it would be or how long it would last for. But I thought we would be able to induce some sort of immune response because nature produces an immune response. People who get coronaviruses become immune. They don't always stay immune for that long, but they become immune. So if nature can do it by itself, then we can emulate that. So I thought there would be a vaccine. What was completely unclear was how efficacious that vaccine would be. I think what has been extraordinary is the speed with which we have developed a product that is now being used, around the world. That has never happened before, and as humankind should be proud of itself for achieving that.
Yeah, and how about the new kind of class of vaccine, which, but wait, maybe first up, kind of like the traditional vaccine of exposing something to a small amount so it can build its own resistance. That's not the approach taken by the big kind of Pfizer and Moderna vaccines, amongst others, is it? What's the difference in that new approach, or is it entirely new?
Well, it's not entirely new because that technology was being developed, but it was in the process of being developed. It had never been used in an approved vaccine. There were signs that approach would work, but we didn't have any products using this technology called mRNA. So it was kind of experimental. It happens to work extremely well. We found that out pretty quickly, amazingly well. And one can understand the theory of why that's so, but until you try something, you just don't know how effective it's going to be and whether there's going to be some horrible side effect or not. But it turns out that it's both incredibly safe and wonderfully efficacious. But there are lots of, there isn't one form of vaccine. We have the oldest type of vaccine are either using germs that are closely related to the disease we're trying to prevent. And the first vaccine ever, the smallpox vaccine, was one of those. Jenna used a cowpox, which is a close relation, but doesn't actually cause bad disease in humans. But there was a kind of immunological cross-reaction. The next kind of series of vaccines were dead vaccines. So you get a germ, you make sure you kill it, and then you inject it, and the body sees it. It's not quite the same as a live one, but there's enough bits on them in there for the body to recognize, and that was the kind of next stage. Then the stage after that was what's called live attenuated, which may be better termed as munted. So what you get is you get a sort of normal germ and you munt it. So it's not quite dead, but it's got a very bad headache. And it doesn't do its job properly, but it's still kind of vaguely alive. And there's quite a few of those vaccines. We give them, and the body goes, oh, look, there's a live vaccine, a live germ. But actually the germ has almost had it. So again, it doesn't cause disease, but and then the next generation is what's called subunits, where we look at the germ and then we just take little bits of it. And this is the whooping cough, the pertussis vaccine is one of those where it's actually sort of 5 different bits of the germ that we just make independently and put that in a vaccine. So it's not the whole germ anymore, it's just little bits of the germ. And then that has gone on to all sorts of iterations where bits of the germ are delivered in various ways. And that's what we're seeing now with most of the candidates that are being used. The bits of the germ, in this case the SARS-CoV-2 COVID-19 virus, scientists take bits of that and put it either into a completely irrelevant virus, or they put it into this genetic material, the mRNA, and deliver these bits by various mechanisms.
And that, when you mentioned before that, the body can see, what is the kind of role of the spatial recognition of the shapes around, the corona shapes around the COVID-19 virus? And how what's the breakthrough with the mRNA in helping the body recognize that?
So, the immune system takes a deconstructed approach. So whenever it sees a germ or a bit of a germ, it pulls it to pieces, basically. And then the immune response is always to smaller bits of the germ, never the whole germ. And the way the mRNA works is that this is a genetic code. So we put the code of a part of the COVID-19 virus called the spike protein into this mRNA coding, and then that is wrapped up in a kind of fatty, sort of lipid fatty kind of coating, and it's injected. That makes its way into cells, human cells, when it's been injected locally, and then the genetic code instructs those cells to make this spike protein. And so the spike protein is produced not in a factory like in the olden days, but actually the body is used as a factory. And this spike protein is made, and then the immune system goes, what's that doing there? That's not part of me. That's foreign. I'm going to react to it, and that's what happens. And it reacts very nicely to it.
Wow. And that kind of ability to program cells to produce things that are kind of alien to them, is that, what are the other applications of that? What are the potential other applications, or is it too early to know?
Well, there are some fairly obvious ones, and these were being developed anyway, as I say, because up until COVID-19, these were experimental, and front runners were vaccines to treat cancer. And then there were other infectious disease vaccines that were also being developed.
And how exciting is it that, how long were the timeframes before for this likely to be, for wide usage? And how fast have things been moved forwards?
It's just extraordinary, as I said earlier. You'd expect development times of 1 to 2 decades in the olden days. And here, within a year, we're injecting people from just learning about the virus. And that is extraordinary. And it's required a lot of different steps. Money. There was a huge amount of money that went into it. And that, of course, made a huge difference. But also the whole regulatory side. And regulation is really important. It's an absolutely essential framework which every country has to make sure that what we give as therapeutics, and including vaccines, are actually safe. Having been through that process again and again, it really is very, very stringent. But up until COVID-19, it has always been a kind of a serial process where you find out certain things and then you send it to the regulatory authority and they have to think about it and they come back to you. And then you have to do a whole lot more steps and then you go back again. And then finally, after some clinical trials and everything, you get to approval. And what happened with COVID-19, that it became much more sort of parallel approach where you say, right, we're doing this and we know this now, but these are in process, and if this works, will you be okay? And all of a sudden, the whole regulatory process has sped up enormously, and that's allowed us to get approvals, even if they're provisional approvals, to allow the use of the vaccine.
Kia ora, I'm Sophie.
And I'm Simon.
And I'm Alice, and together we host the Spin-Off's food podcast, Dietary Requirements.
Join us each month as we explore a vast culinary landscape, from the gourmet
Ooh la la
to your more hearty tucker.
Kiwi onion dip, anyone?
Everything's on the table in Dietary Requirements.
Subscribe wherever you listen to all your other favourite podcasts.
And so when you started the CVC company to try and take a bit of a different approach, you started with two approaches, didn't you? And then you were surprised by the speed with which the developments happened overseas.
I don't know if we were that surprised, but again, we didn't. What we were surprised about, what we were all surprised about, certainly me, was actually the degree of efficacy of the mRNA vaccines, which were the first ones to read out. Yes, we started with 2 programs. And the first program was to do exactly what everybody else was doing, but with our different delivery systems. So I talked about isolating this spike protein, which is from the coronavirus. If you've seen pictures of the coronavirus, it's a sort of round ball with things, with sort of spiky stuff sticking out. That the spike protein, the thing you see on all the little graphics. And we took the spike protein, and the idea was to put that onto these biodegradable biobeads, which were the technology coming out of Palmerston North and Polybatics. And that was to achieve the same thing that everyone else was trying to achieve. But also at the very beginning, and this was primarily my concern, is that I was concerned that we would see what's called escape mutants. So one of my worries was that everybody, including our first approach, was using one bit of the virus. And every type of life form, including viruses, always try to work around any objects that are in their way. So if you have an immune response to a spike protein, the virus will evolve, if it can, to get around that. And that's what I'm calling escape, and what we now call variants. And I was always worried that would happen. And so we developed another parallel approach, and that was to take all sorts of bits of the virus, not just the spike protein, but various other bits as well, and package them up into a rather more complicated type of vaccine called a cytotoxic T-cell vaccine. And that's designed to do something a little different from produce these neutralizing antibodies, which seems to be the main mechanism for all the other vaccines. And this type of immune approach is actually harder in a lot of different ways. But what it aims to do is, rather than to prevent the virus entering a cell in the first place and sort of blocking its entry, which is what these neutralizing antibodies do, this approach is designed to recognize specifically just cells that are infected with the COVID-19 virus and deactivate and kill those cells only. And that's cyto means cell, and toxic means toxic kill. So we're trying to specifically wipe out those cells that get infected with the COVID-19 virus. And as I say, that was specifically because I was always concerned that we would see the virus trying to get around the approach that everybody's using at the moment.
And how have things been going for you in the development of that other approach?
Yeah, so we started in April and May, and we were doing this spike protein thing, and then in kind of November of last year, the results came out from the mRNA, the Moderna one first, and we thought, wow, that's fantastic. We don't need to move forward on that anymore. So we actually immediately stopped that program. We thought, well, the biobeads were a nice idea. Maybe they would work, maybe they won't, but something else already works. That's fine. So that nut is cracked. But we remained concerned, and we still didn't see the variants at that stage, but we were still concerned that the variants would happen. So we continued with the what's called the cytotoxic T-cell approach, and we kind of broadened the program a bit. So we said, well, we've got these biobeads, so we'll try those, but we'll also try the same type of vaccine without biobeads, and we'll just find out what works best. Yeah. So that's what we're doing at the moment. So hopefully, by within the next 6 months, we're going to have quite a lot of data which will show us whether or not we are seeing a good T-cell response.
First up, talk us through the what the biobeads are and what they do. What's the new thing there?
So it's quite neat. You always want to deliver your vaccine in a way that tickles up the immune response as much as possible. And one way of doing that is to administer your whatever vaccine you're doing in a form that the body thinks is a virus, because bodies, our immune system are designed to detect viruses. And so if it sees a little round particle of virus size with the repetitive nature of something on the outside, it says, that's something foreign, I need to react strongly to that. So these biobeads are actually produced spontaneously in bacteria. And they are about the size of a large virus. And what's neat is that you can program a bacteria to produce not only these little biobeads, and I'll say what they're made of in a minute, but they are automatically coated in whatever we tell them, in this case, the bit of the Covid-19 virus. So all in one go, the bacteria produces not only this biobead, but also the coating of parts of the virus. And we expect, we'll find out soon, that the body responds strongly to that. The biobeads themselves are made out of a biodegradable product, which has actually been used for decades in dissolvable sutures. So we know that it is harmless. They just dissolve away. So it's quite a neat little system that we will try.
Do you feed that material to the bacteria? Or
No, the bacteria make it all by themselves.
They make it all by themselves.
Yeah.
Wow, that's remarkable. Yeah. And then in terms of, and the second part of the question there is then, how do you go about the testing process? And, at what stage do you get people involved? Or, how does that track?
Yeah, well, kind of people... Once you get people involved, you're talking about spending many, many million, tens and hundreds of millions of dollars, and we don't have those resources. So all our testing at the moment is in the laboratory, and that's all underway at the moment. And what our strategy is to test after test after test and demonstrate, we hope, that our approach is actually something that's very sensible and works, and we want to produce a data package and say, look, this is interesting stuff. The world needs this because we need to be worried about variants, and this is a solution. And once we've got that data package, I'm going to be approaching people all around the world, companies around the world, and saying, look, would you like to acquire this? Would you like to work with us? Would you like to partner? Whatever, to move this forward. And hopefully at that stage someone is going to say yes, and then we would move forward on that basis, and the funding would come from outside New Zealand.
Yeah, and that approach, the cytotoxic T-cell approach, is that really a unique approach that you're spearheading here?
No, it's not. It's not. First of all, some quite conventional vaccines will include some sort of cytotoxic T-cell approach. Making it deliberately is a relatively new idea. There are people around. We're not the only people doing it. People are also trying various other, constructs of viruses, but not many. We're actually one of quite a few groups that are doing this approach, and our particular way of doing things is certainly unique. So we'll see.
And how does it help with tackling these variants? So as the variants arise, are you able to then put that new information in as the kind of the things to look for in terms of the kill?
Yeah. So the whole idea of ours is that we don't need to change a vaccine to cope with these variants, because what we're doing is taking a very diverse set of different bits of the virus. So we've got over 35 different bits of the virus, all kind of stuck in our particular construct. So far, the last time I looked, of all the variants of concern, there's been no mutations in any of our little bits so far. But even if there were, there's so many other bits, there's a redundancy there that we're still, covering the vast majority of possible variants. So it's a kind of diversity approach that is supposed to overcome the variants. And it goes back to what my original concern when I say, previously the vaccines are based on one target, whereas our vaccine is based on a whole set of different targets.
Yeah, wow. So even as it mutates, there'll still be enough in common that you'll still be... Yeah, and, as an expert in the field, do you look at the numbers in places like America and India, and go, oh boy, there's going to be a lot more variants coming out? The Spanish flu, where some of the mutations actually meant that it was less lethal and less of a concern.
Well, you just don't know.
Yeah.
You just can't predict that. So we'll just have to see. So far, it seems to be going in the direction of increased infectivity and certainly no decrease in severity. If anything, an increase in severity. Maybe more severe disease in younger people, but we haven't seen any signs of lesser disease yet. That may happen. Who knows?
In terms of building the company around this and how you've managed to chart it, has it been easy to get kind of funding and support in this environment, or is it always a challenge? Or how do you go about building a business around this?
Funding is never easy. Anyone who says funding is easy is not telling the truth. But we were topical. We had a good story, and most of the funds that we have raised have been through private investors. And I cannot sing their praises enough. It's amazing that various Kiwi investors have been willing to back us with dollars. We've raised a total of $4.3 million from private funds. That's a really significant amount, and that's the only reason we exist. And it's the only reason we can move forward, and it's the reason that we will get to the point where we can say whether or not our ideas work or not. So never easy, but the people backing COVID-19 Vaccine Corporation are just extraordinary. So I cannot thank them enough.
And 4.3 million, I imagine, is a very small fund in the scheme of other virus vaccine production efforts.
Indeed. And, we try and be as efficient as possible. And if we had more funds, we'd be going faster. But, we've got what we've got. We've got a relatively small amount of government funding as well. We're up to about 5 million in total. So, as I say, primarily privately funded. And we'll see how we go.
Yeah, it must be a very interesting time to be an expert in the vaccine area.
Well, I love it. I really love it. It's my medical background. It's my scientific background. It's my commercial background. So it sort of pulls it all together, and I kind of feel I know the field. So it's a very comfortable place for me to be. It's also challenging. Every day. I don't know how much you can imagine this, but when you're on the edge of science, which is where we are, there are always problems, and finding the right people to do each step is always challenging. Sometimes we can manage to find experts in New Zealand to help us. Sometimes we have to go overseas, and sometimes things go wrong. Sometimes things get stuck in customs, and all sorts of little trivial things sometimes can sort of mess you about. Maybe we can't. We do have glass vials, but sourcing the glass vials was a problem for a while, and so all sorts of challenges. Some trivial, as I say, like the customs. Others really technical challenges. We suddenly find something. How on earth do we do this or solve this technical problem? But always we do in the end, and I like those challenges.
And a place where decades are happening in years must be extremely stimulating.
Oh yeah. I mean, yes. I'm very happy to be doing this. It's not without effort, but there is a reward to it as well. And I want to be straight. I don't know whether what we're doing is going to work at the moment. There's a good theory as to why it will work. The science is sound, but until you try it, you don't know. It might just not be good enough. There might be something we haven't thought of yet. So it's always, in the next 6 months is going to be quite interesting because we're actually going to get some readout. So we'll see how that goes. Of course, I hope it's going to be fantastic.
Yeah, and you've pulled together the chair of your company, the managing director of Douglas Pharmaceuticals. That's another great New Zealand success story that, it's quite little known here, I think, that we have a big pharmaceutical player working domestically.
We do. We actually have two. Yeah. New Zealand certainly punches above its weight in the pharmaceutical field. So what is unique about Douglas Pharmaceuticals is that it does all its own manufacturing. I don't know if it's all, but certainly they have a major manufacturing plant, and here in Auckland, and that is amazing. And it's also amazing that Jeff Douglas, who is the managing director of Douglas Pharmaceuticals, has been willing to be our chair. I always try and surround myself with people who really know the field and what they're doing, and Jeff Douglas certainly does, and it's an honour to have him on board.
And what will, as a kind of final question that we always like to ask everyone, what will success be for you personally, and also what will success be for you with CVC?
So success is if I see a construct or a derivative of the construct that we are, working on right now, being injected into humans. That is success for me, and it working. That would be extraordinary, and we shall see.
That's magic. Well, thank you so much, Dr. Robert Feldman, the CEO and founder of CVC, for joining us today to share the story, and really excited to see where you go next.
Pleasure being here.
Thank you very much to Tina Tiller for producing, and thank you very much for having us along and listening. Cheers.
You've been listening to Business is Boring, presented by Simon Pound. And brought to you by The Spin-Off and Callaghan Innovation.
From the Spin-Off Podcast Network, that was Business is Boring, brought to you by SparkLab. Make sure you're following Business is Boring wherever you get your podcasts. And for more information on SparkLab, visit sparklab.co.nz. Ready to rediscover the joys of cycling? With over 300 kilometres of cycle paths across Tāmaki Makaurau, jumping on your bike and going for a ride is such a fun way to discover the city from a different perspective. Cycling is getting more and more popular across Auckland, so now's a great time to join the hype and give cycling a go. Head to at.govt/cycling to find your nearest cycleway today. The Spin-Off Podcast Network.